And thank you to Dr. Gonulu and Dr. Marek Kubicki for hosting this session. And to all of you for coming to this today. I know it's a holiday, and so it's a national holiday, so I appreciate you taking time with us. So for about 45 minutes, I'll take you on a guided tour of different neuroimaging studies that are going on around the world in the field of psychiatry. So many of us are collecting brain images from patients with psychiatric conditions, brain images that reveal the structure, the function, the molecular content of the brain, and there's a lot of interest in relating these to genetic and blood markers in these same conditions. So one approach to analyzing brain images is to use biobanks. So the ENIGMA consortium has published since 2009 the largest neuroimaging studies of about 19 different brain disorders. The goal is to find factors that help or harm the brain by analyzing brain scans and genetic data from a little over 100,000 people from 45 countries. And this has led to the largest neuroimaging studies of schizophrenia, bipolar disorder, major depression, PTSD, substance use, and sleep disorders, also anorexia, OCD, anxiety, conduct disorder, ADHD, and NASD, and a number of neurological conditions. So the analyses of all these brain scans has two main goals. One is to decide how the major disorders of the brain affect the brain, at least as we can see from MRI, diffusion imaging, resting state fMRI, EEG, and MEG, but also to ask how does genetic variation affect the brain. So ENIGMA screens millions of markers across the genome with different methods to find genetic markers and also environmental factors that affect brain development and aging and disease risk and clinical outcomes. So a summary of today's talk is, I'll tell you what is ENIGMA, how do the neuroscientists around the world work together. We'll talk about worldwide genetic analysis of brain images, so how to link neuroimaging with genomics. We'll talk about global studies of brain disorders and some of the challenges and advantages of pooling imaging data internationally, and then some fun new directions using AI and looking at interventional methods. So this is where the data is. People are scanning patients every day in these locations. The color is the disorder that they're studying, so there's a key at the bottom. And ENIGMA's data isn't in one place, it's all over the world. And there's various manuscripts about how this came about and how the scans are analyzed. And about 70 journal papers are published every year by ENIGMA, and there's even a special issue of these papers. So the disorders studied span psychiatry and neurology. If you look on the top left, there's bipolar disorder, schizophrenia, major depression, also studies of early onset psychosis, people at clinical high risk for psychosis. At the bottom, there's obsessive compulsive disorder, autism, Tourette, ADHD, which share some symptom overlap. In the middle, there's neurological conditions such as epilepsy, ataxia, stroke, and Parkinson's. And of course, these aren't homogeneous. So someone with a brain injury may have incurred it in the military or playing sport or as a child. So there's subgroups within these groups. And then there are technical groups. So the data collected might be diffusion MRI, EEG, might be genetic data. So special groups organize the analysis of these types of data for the clinical groups. So that's just a map of the different disorders that are studied. There are new groups on chronic pain, on migraine, on neuromodulation, meditation, and also pollution, how pollution affects the brain. And these are some of the people that lead these efforts. So let's say you're a scientist, which is a little black dot, and the other dots are the people you work with. You can imagine that for different diseases, there might be groups of scientists analyzing structural images of the brain, diffusion images, functional images, and they can work across disorders, or they could work across types of brain imaging and put together the different pictures that they get of the various diseases. And these are some examples. So these are 9 psychiatric conditions: major depression, bipolar disorder, schizophrenia, alcohol use disorder. And these are the patterns of gray matter thickness reduction in the different disorders. And I'll take you through some of these findings in a minute. And you can get a t-shirt if you want to wear your data, you can get one of these t-shirts. So for those people that don't do neuroimaging, I'll tell you what you do to analyze the data. So you would collect a brain scan, maybe a 3D structural brain scan, or a PET scan, which might look at brain glucose metabolism or amyloid or some molecule. And then using techniques such as SPM or FreeSurfer, you would measure the thickness of the gray matter, the volume of white matter, maybe CSF, and you could make statistical parametric maps. You could say for patients with this disease here, Alzheimer's disease, where is there evidence that on average the gray matter volume or the gray matter thickness is reduced in Alzheimer's disease? And of course, in Alzheimer's disease, the volume of gray matter is about 2 standard deviations lower than healthy controls, and this is matching the pattern of beta amyloid that builds up in the brain. And this is what the data really looks like. If you had a patient with Alzheimer's disease and they came back to the scanner every year, you'd see progressive atrophy. And then if you look at the different sections of the brain, it's not totally obvious where the cortical changes take place. So if you look at let me see if I can get this to work. Oh, it doesn't work. The other one worked better. If you look at the cortex, it's difficult to appreciate where exactly the tissue is lost. And so you can imagine that's why some of the image analysis methods are used. So if, for example, you were interested in tracking a patient over time, and this again is Alzheimer's disease, a neurological condition, you could plot at every month what is the sequence of gray matter loss. And there's this sort of trajectory, this is Alzheimer's disease, of early gray matter loss in the temporal cortex, moving into the parietal cortex, eventually into the frontal cortex. And in the interior of the brain, you see the limbic system, the cingulate gyrus is affected by gray matter loss in a stereotypical sequence. Now, if we turn to schizophrenia, these are patients with early onset psychosis, and the gray matter loss is mapped. And you'll see that it's a little more subtle. It has a slightly different pattern. And we're going to look at some evidence that this is actually a composite of many subtypes. So it's not true to say every patient will undergo this change. There might be different subtypes that we can see within there. But one other thing is you can use these in drug trials. And so if you have patients taking second-generation antipsychotics, such as olanzapine, or patients taking first-generation antipsychotics, such as haloperidol, you can ask where it is in the brain that gray matter progression of loss is less in the second-generation antipsychotic treated group. Now, does anyone know what disease this is? Can anyone guess what disease this is? It's not a disease. You know the answer. So the problem is this is normal aging. So the changes that you see here in development, primary sensory motor cortex is changing early, and then association cortex in the stereotypical pattern. And so what you would really want to do is have a normative model of the normal changes. So if you have healthy people from age 0 to 80, this is the volume of the thalamus, and it goes up. When you start university, it starts to go down. And then there's a more precipitous loss of volume around age 60. So these centiles can be used in an approach called normative modeling to place your patient relative to the percentiles for that brain feature in a healthy reference population. And this is work by Sophia Frangu in the Lancet Digital Health, where each dot is a patient. I think this is hippocampal volume. And the hippocampal volume varies by a factor of 2 among healthy people. There's a large range. But you can see each color is a different cohort of people who have contributed to the study. And obviously, you can make charts for different structures. The different parts of the brain have different maturational trajectories. So when you have a patient and the measure is being compared to these references, obviously what we expect for the amygdala is very gentle loss, whereas for the thalamus, there might be faster loss happening normally in healthy people. And you can look at infants. Rebecca Nickmeyer leads ENIGMA Origins. She scans little babies. And essentially from age 0 to 6, the boys are in blue, the girls are in red. This is based on 3,000 scans. There's a very characteristic pattern with quite an early plateau. I don't think they stop at the age of 6, but it looks like there's a gentle plateauing of, in this case, intracranial volume. That's the overall size of the brain. So, and this is work by Ann Alex. It was in Nature Neuroscience. She found using this method that infants that are born prematurely catch up in their brain growth. It's a very interesting use of these. So despite preterm growth, there was a catch-up with healthy children. So you could ask, I mean, maybe you're studying eating disorders or schizophrenia. You could study if a patient is relapsing or remitting relative to the other healthy people of the same demographic using this model. And you could use a so-called Z-score or Z-score that says where is the patient within this distribution of healthy people. And then this might be something you could plot for each patient as they're receiving a treatment or as they are undergoing some other intervention, maybe TMS. And this is brain structure. You could do this for any measure. It could be functional activity, or it could be dopamine receptor binding or something else. Now, one problem is each site collects data with slightly different characteristics. And so if in color there you put data together from different centers around the world, they don't nicely align. So there's a method for so-called site correction, and this is a nice review about how to do that, where at your site you scan some healthy controls, and then the range of data from your healthy controls is shifted to match the reference curve, and then you can use the reference curve as a percentile system. So this is called centile brain. You can use it. This is hippocampal volume, actually in women. There's different charts for women and for men. So you can say, okay, let's use these charts to decide if anything is affecting the brain. So the first one might be genetics. So 10 years ago, a gene was discovered, TREM2, where 1% of us have a mutation, a polymorphism, and it increases your risk of Alzheimer's disease by a factor of 2. So you could say, okay, does that mean that people with TREM2 lose tissue when they're old or when they're young? I mean, maybe we can intervene in this. And this is useful for drug trial enrichment, because if you could find people who are likely to decline faster, if they have a genetic makeup that makes them decline faster, you want to enroll those people in drug trials. There's no point enrolling people that are healthy and are going to stay healthy in a drug trial, because you won't find an effect of your drug. So one thing ENIGMA does is to find genes that help or harm the brain. So normally you can sequence the genome, look at the different letters or nucleotides in your DNA, and you can ask if there's any statistical difference in the genetic code between patients with a certain disease like schizophrenia and healthy controls. And that's the normal way of finding genes associated with a disorder. In imaging genetics, you can do the same thing with images and genes and say, is there anything in the brain that is affected by the genes? And so this was not very popular initially. So this is Nick Martin. He said, if you want to discover genes that affect the brain, you will need brain images from 50,000 people. Just because brain images are more expensive to collect than measures of height or weight, it doesn't change the power calculations. But if you think of the movement of biological psychiatry, so finding endophenotypes or finding biomarkers, we think that certain biomarkers of disease might have a simpler genetic architecture. Maybe there are fewer genetic variants. Maybe the genetic variants have a bigger effect. And in neurology, that is true. So there are mutations in the amyloid precursor protein that lead to amyloid to build up in the brain, and that leads to brain atrophy and cognitive decline. So maybe in psychiatry, if we could find a good imaging marker, we could do genetics on it instead. But it's a hypothesis. It's a hypothesis. Maybe this isn't any better than doing diagnosis. So the first ENIGMA study measured the size of different parts of the brain. It did a genetic screen. And if you're not used to seeing these plots, they're called Manhattan plots. And on the x-axis, there's the genome, chromosome 1, 2, 3, 4. And then the height of the y-axis is the evidence that that marker in the genome is associated with the brain measure. And so there's various things called genome-wide significant hits. They're markers where people with a certain genotype have bigger or smaller brain volume. And you can basically march through the genome and find areas of the brain that the genes actually affect. And if you do this for Alzheimer's disease, the biggest hit is ApoE. So 1/3 of us carry ApoE4. We have 3 times the risk of getting Alzheimer's disease in our life. But there are lots of other hot spots on the genome where if you have a single letter change, your risk for Alzheimer's is about 10% higher or 20% higher. And you can basically say that these are overrepresented in Alzheimer's patients. These genetic code variants are overrepresented in Alzheimer's patients. And there's about 200 of them. If you add up each of the genetic variants that gives you higher risk for Alzheimer's disease, and if you weight them by how risky they are, you can get a polygenic risk score for Alzheimer's disease. And your risk as you get older of getting Alzheimer's disease becomes almost certain. And this was developed by a radiologist called Raul Desikan, who very sadly passed away. He had ALS, but he was one of the pioneers in doing this type of work. Now, in psychiatry, the genetics are more complex. There are immune and inflammatory genes, as Marek and Lauren and Johanna talked about. Inflammation in schizophrenia is implicated. Dopamine receptor subunits are implicated. And as you collect more and more genetic data, more and more genes are implicated in schizophrenia. So you could do this with brain images. You could say, can everyone please measure the size of parts of the brain? Or could you get diffusion tensor imaging and measure the microstructure? And then we're going to test for associations between the genetic code and these measures. And then we're going to do a meta-analysis. So if someone collected 10 brain MRI scans, but someone else collected 10,000 brain MRI scans, we're going to meta-analyze the evidence. And so this led to the discovery of apoptotic genes where polymorphisms affect the hippocampal volume. The same gene, the harakiri gene, which variation affects in that gene affects the hippocampal volume, was also found in Dagestan. This is a group of people in the Caucasus Mountains. Their doctor is Kazima Bulayeva. She's a Russian geneticist. And she found that the same region of the genome that affects hippocampal volume confers risk for mental retardation in the reason they study this group is there's a lot of inbreeding. So it's very high mountainous area. There's a lot of runs of homozygosity, or essentially genetic inbreeding. And if you have a mutation in that gene in one chromosome, there's lower hippocampal volume. But if you have a mutation in 2 chromosomes, it tends to lead to mental retardation. So this can be done to look at the effects of ApoE, the Alzheimer gene. And that doesn't matter if you're young, but when you're old, your hippocampal volume is 2 standard deviations lower if you carry the ApoE4 variant. And you can keep doing all these things. I just want to show you that this works, and so you can apply this to every brain measure that you care to look at. You can even, for each part of the brain, make a polygenic score. So you could say all of the letters of the genome that are associated with the volume of that part of the brain, let's add them all up, and we'll predict brain volumes from these. And it's pretty amazing. You can predict about 10% of the variation in subcortical brain volumes just from the gene sequence by adding up these different letters. And this has been shown in people of different ancestry. Now, as you move around the cortex, the genetic loci that affect each region's cortical thickness are partially overlapping and partially distinct. So you can imagine there might be genetic programs that influence the development or aging of different cortical sectors. And this, in fact, was found. So this paper in Science, The Genetic Architecture of the Human Cerebral Cortex, it found lots of genes that affect the cortex, in fact, a huge number. But then they were grouped. So the ones that affected a certain sector of the cortex were very similar, and another block seemed to be affected by a second set of genes. So you can think of genetic modules that might exert their effect in a sort of tiling pattern. So you can do this. Oh, I should mention genetic deletions. So some people have an expansion of the genome, like in Huntington's disease, there's trinucleotide repeats, or in some children there might be a deletion, like 22q11 deletion syndrome. And these have huge effects. So these are a little bit different. In ENIGMA, we do look at them. There's not many people with these rare variants. But you can make a catalog of how each rare variant affects the brain and ask if the effects on the brain are similar to the diseases that they confer risk for. So some of these genetic deletions lead to increased risk of schizophrenia, of autism, and you can ask the question, is the pattern of anomalies like those diseases? And in fact it is. So this work by Kuldeep Kunar and Claudio Modinato, they've been looking at genetic deletions and clustering them with idiopathic psychiatric conditions. and finding that some of the conditions like 22q11 deletion syndrome resemble schizophrenia, which is something that is higher risk in these people. And this is an ongoing and interesting project where they cluster the deletions with the idiopathic disorders. And this gentleman, he's rather imposing. He runs the Decode Genetics Iceland Biobank, and they have the biggest data set of genetic deletions and brain scans in the world, and you can go and see him. It's very nice there in Iceland. So that, I won't tell you much more about genetics. I mean, there's a lot of cool things. One last thing maybe is epigenetic changes in the genome, methylation, are being studied by Sylvain de Rivière at King's College London. And she has found that methylation of homeobox genes also predicts regional brain volumes. And there's no doubt about this. It's been replicated across 11 different cohorts. So that's kind of a cool study. So let's look at the psychiatric illnesses. So let's look at some of the imaging data for the conditions that are listed here. So ENIGMA members, when they're looking at psychiatric illnesses, will look at MRI, DTI, they'll process the data. But this time they're going to test associations between brain measures and disease, either diagnosis or not, or particular clinical measures or treatment. And then we're still going to do meta-analysis. So we're going to combine all the data across sites, and if a group has scanned a small number of patients, their contribution will be weighted a little bit less. And in ENIGMA schizophrenia, all the sites scanning here, there's maybe 4,000 patients with schizophrenia, I think a total of 33 sites. And they ran this parcellation program. Anyone can use this. It's called FreeSurfer. And they asked whether there was evidence that the gray matter thickness was lower in patients with schizophrenia. Now, you all know that every patient is different, so this is an average. But in 25 of 33 cohorts, they found evidence for thinning, in this case of the fusiform gyrus. And one of the things that was interesting is every part of the brain except the visual cortex showed evidence for a reduction in schizophrenia. And I want you to look at the effect size. So what's happening is Cohen's d is the difference between the two group means. So the average of controls and the average of schizophrenia patients are going to be subtracted. And we're going to measure that like in a normative model by saying how many standard deviations lower are patients. And patients are about minus 0.5, or they're about half a standard deviation lower. And you'll remember that in Alzheimer's disease, they were 2 standard deviations lower. So the effect in schizophrenia is about 1/4 of that in Alzheimer's disease. But are there subtypes of schizophrenia? So you can subtype the patients based on, number 1, clinical definitions, or number 2, data-driven clustering. And there's different methods that you can try for this. So you could say, well, some patients with schizophrenia have very severe depression. In fact, there's something called, many of you will know this, deficit schizophrenia, where a third of schizophrenia patients have primary enduring negative symptoms and poorer prognosis. And this group, led by Nerissa Benage, found that they have much greater cortical thinning if they also have major depression than patients without deficit schizophrenia. And there's a sort of stereotypical pattern where they have poorer prognosis and thinner cortex. The other way to do this is to not use clinical data, and you just look at the images to find clusters. So this is a study by a Chinese consortium that was working with ENIGMA. And I think Dr. Gonal and I really like this method. It's fun to try. This method is called SUSTAIN. So what it does is, I'll tell you the result first. They found that when schizophrenia progresses as the patients get older, there's a group of patients that have frontal first progressive gray matter loss, and there's another group of patients that have temporal lobe first gray matter loss. And what they did is they took 4,000 patients, and using SUSTAIN, they split the patients into 2 different groups, subtype 1 with frontal and subtype 2 with temporal atrophy. This is work by Alexandra Young. She's at University College London, and she wrote the software to do this. Now, how do you do this subtyping? What you do is for every brain measure, you say, when does it become abnormal? When does its Z-score or Z-score go lower than usually 2 standard deviations below the controls? And you sort them into an order. You say, well, the frontal lobe seems to become abnormal later than the visual cortex. and you put these all in a rank order. Now, not every patient will be the same, so it tries to make a consensus about what the sequence of abnormalities is, and then after that, it divides the data to make better clusters. And this is interesting. I mean, you could imagine that all the disorders we study might be a mix of different subtypes. Maybe they're not subtypes, maybe they're dimensions. I mean, it might be like soup, and soup has a lot of ingredients, and there might be different amounts of the ingredients. So rather than think of these as distinct entities where each patient falls into two categories, maybe there's a mix of these. But it could be that medication has a very good effect in one subtype but not in the other, and this would be a more refined way of understanding what's going on. And this was found in Chinese people and also in European ancestry people, and the subtypes are basically the same. In fact, the frontal first subtype seems very similar between Asian and non-Asian populations. You could ask, do people who develop psychosis have changes in the same regions that change first in schizophrenia? So this is Maria Jalbrikowski and Dennis Hernaus. They lead ENIGMA's high risk for psychosis group. And it is not entirely true. So the regions that seem to predict psychosis onset include the paracentral cortex, which would not probably be what people would hypothesize. But this is a way of doing prognosis using the same data in clinical follow-up. You could ask if the laterality of the brain is different in schizophrenia. This has been a hypothesis for 50 years, and the answer is not much. It's very slightly less in schizophrenia. So let's look at the different disorders. Here's major depression. All these are cortical thickness maps, bipolar disorder, schizophrenia, all based on very large international samples. And you can start to see that there might be some patterns. So this is gray matter thinning in different psychiatric conditions. Some of them primarily neurodevelopmental, like OCD, autism spectrum disorder. Notice how autism spectrum disorder is the only condition where there's more gray matter. It's very puzzling. There's more gray matter in cases with autism. All the rest, there's gray matter deficits, except the deletion syndromes. The deletion syndromes are an exception. There's some higher, some lower gray matter. Which disorder do you think in psychiatry has the greatest effect on the brain? Well, there's a few people here that study this. So in anorexia, this study is by Esther Walton and Stefan Ehrlich. One standard deviation lower than controls, and this is the average of patients with anorexia. And as Lauren Breithaupt discussed yesterday, there is some evidence of at least partial recovery in the gray matter in a subset of patients. So, I mean, that's a really interesting case in psychiatry. Now, let's just look at the more common disorders. So major depression tends to at least structurally affect the limbic system. Bipolar tends to affect the frontal cortex, so the inhibitory deficits lead to mania, or they're associated with mania, and this is very distinctive between those two conditions. You can also do voxel-based morphometry. So when you do voxel-based morphometry, you don't just look at the cortex, you make a map of the disease effect on the whole brain. This is a huge study of bipolar. And as well as the frontal deficits in bipolar, you see some evidence in the back of the corpus callosum, the splenium. It's quite interesting. It's an association commissure, which has very large effects, differences between bipolar patients and controls. Now, you have to be a little bit careful. So all these patients aren't the same. So some people with major depression have been taking antidepressants, some haven't. Why do you think the people taking antidepressants have poorer looking brain structure? Well, it's not a randomized clinical trial, so people don't randomly take medication. The people that were more severely ill or ill for longer or had an earlier onset tend to have more medication. So if you use a biobank to look at medication associations, you will often get an effect that's the wrong way around because it's not a randomized clinical trial. Now, we have to do this, but it's not a good way of checking how the medication affects the brain. It's cross-sectional. So you could say, well, give me a readout of the different structures that best differentiate the different psychiatric conditions. You'll always find the hippocampus is one of the strongest effects. So in schizophrenia, it's about -0.4 or about half a standard deviation too low. In bipolar disorder, it's about -0.2 or about a fifth of a standard deviation too low. And even in major depression, you'll see some controversy about whether there are hippocampal effects, but they tend to be consistently found. What are the modulators? So not every patient has the same degree of structural atrophy. A big factor is cardiovascular health, and so L'Oreal Dietz and Thomas Hayek in Nova Scotia argue the following. They say that if you look after your cardiovascular health, which is not easy if you have major depression, but they argue that BMI, body mass index, and a number of cardiovascular factors interact in a synergistic bad way with the effects of the disorder. And so they're very interested in examining the effects of cardiovascular risk. And they think that BMI potentiates the bipolar disorder effects and interacts with it, and that they're not independent, so that cardiovascular treatment, semaglutide is a new one for weight loss, may be of interest in these different conditions. Now, there is interest in different medications. So if you look at lithium or antidepressants or antipsychotics, or for that matter, anticonvulsants, they all have a known mode of action. If you look at what modulates the disease effects, patients with bipolar disorder taking lithium tend to have gray matter hypertrophy. Their gray matter looks thicker. Patients taking anticonvulsants, which are anti-seizure medications, have much lower. And Chris Ching at USC leads the Bipolar Medication Initiative, where he argues that the large medication effects in bipolar disorder make it very hard to disentangle the effects of the disorder. They often have medications with multiple modes of action, or polypharmacy, people that take multiple medications. So they're recoding medication histories using the ECNP's neuroscience-based nomenclature. And for those of you who are not familiar with that, you code the medications based on their known mode of action, which may vary by dose. And then you group the patients taking these different medications and then look at the brain maps. So this sort of leads to a different perspective. If you look at brain volume differences in bipolar, you go, oh, the hippocampus is too low, the ventricles are too high. But if you split by medication, it is extremely complicated. So lithium, increase in gray matter, anticonvulsant, lower gray matter. It's a very, very, very complicated picture. Moving to childhood disorders, what is the childhood disorder with the greatest effect on the brain? So this is Marlena Staginas, and she studies antisocial behavior and conduct disorder, people who are aggressive, they have externalizing symptoms. And she found that people with conduct disorder have the greatest brain differences compared to ADHD, where the brain differences are also frontal, and very, very small effects in anxiety and major depression. And this is all children, so you'll see that there's a sort of difference where externalizing symptoms, which are prominent in conduct disorder, tend to link with frontal lobe deficits. Now, we had a wonderful session yesterday on diffusion imaging, so I'll try and do a good job of diffusion MRI with experts all here. You can collect diffusion imaging data that measures the spread of water diffusing in the brain. It's not isotropic, as we've seen when fibers are crossing or when myelin is degrading. All these biological processes affect the diffusion signal. And these are diffusion tensors. They are directional. There's a lot of very cool stuff going on. That's the corpus callosum up there at the top. And you could use these to track fibers, and I mean, there's a lot of wonderful things that you could look at using diffusion MRI. So you could say, well, what if ENIGMA looks at these diffusion measures, FA, radial diffusivity, mean diffusivity, and you heard Johanna and others yesterday talk about free water, which is a very interesting measure. And they all characterize different aspects of cellular processes that influence diffusion, either myelin, inflammation, we heard about yesterday, cell loss. And this is done in ENIGMA using a tool called TBSS, or tract-based spatial statistics. And people basically studied all the different diseases with DTI. Researchers from all over the world were taking the lead on these projects. And let's have a look at some of the results. So do you think the effects of schizophrenia on DTI will be larger or less than just good old structural imaging? It's about the same. And we weren't even using a good DTI measure. So the Boston gang is using a better measure than we were, free water. But you'll see that fractional anisotropy differences in schizophrenia, which is a measure of the directionality of diffusion in the brain, nearly every site with schizophrenia patients found that that was altered. It was not controversial at all. In fact, this is a forest plot. I mean, every single site, I don't think there's any exception, maybe one exception had difficulty finding that. So the DTI effect size in schizophrenia are about a Cohen's d of minus 0.4. That's about half a standard deviation too low. It's not to say that there aren't better diffusion measures. In fact, free water that we heard about yesterday is probably better than this. But these anomalies are much less affected by medication. We find that lithium, antipsychotics, anticonvulsants, at least in our studies, don't affect these measures very much at all. Meanwhile, in Japan, the Japanese members of ENIGMA formed a consortium called COCORO, and they found that all of the rank order of the brain measures in terms of how abnormal they were in schizophrenia and bipolar and depression, the rank order was essentially the same. So if you're in Japan and you have schizophrenia, the brain anomalies are very similar to if you're in Turkey or Western Europe, or for that matter, in other parts of Asia. The only problem is the modulators of these effects, there are three main ones. So one of them is duration of illness, longer illness, worse brain abnormalities. Age of onset, which is kind of the same as duration of illness, or it's correlated. Earlier age of onset, worse brain abnormalities. And proportion of the cohort that's medicated. And you'll remember there's this weird effect where if a cohort is receiving more medication or for a longer period of time, the brain deficits are worse. But it's a confound. The patients with worse symptoms get more medication. So you'll always see that a patient cohort taking more medication tends to come out worse. You can do normative modeling with diffusion. So ENIGMA has a normative model with 52,000 people's diffusion measures. The problem is that diffusion has a very large site effect. And so if you have big voxels, the FA goes down. So you need to use things like combat, which is a method to adjust for scanner and protocol effects before you can do normative modeling. And you can make lifespan curves. They're kind of interesting to do for diffusion. And you can plot patients on these normative curves. So this is a strange normative curve. So rather than going up and then going down when you're old, mean diffusivity, which is how much water diffusion there is on average, that gets low and then goes up. So when your brain deteriorates, there's more diffusion. And you can plot, these are Parkinson's disease patients. And there's really strange results. So in neurology, you'd assume that all of the white matter changes are negative. But in early Parkinson's disease, stage 1 of Parkinson's disease, the metrics appear better than normal. But thinking about what you heard yesterday, there may be some inflammatory effect that is going on in there. So now this is all phenomenology. I mean, we're looking at brains. It's like, I mean, what is the mechanism? I mean, we talked about genetics earlier. Is there sort of a mechanism behind these? So one approach is to correlate the brain maps that come from MRI to gene expression maps. So let's say there were genes that were only expressed in neurons, and there are. And let's say there's different genes that are only expressed in glial cells. And there might be genes like myelin basic protein or proteolipid protein that are only expressed in myelin, or very, very much more strongly expressed in myelin. So some people don't like this approach, but I'll tell you what it's called. It's called virtual histology. So you take an imaging brain map, and then you line up the expression of a gene, or you line up a map of myelination, and you look at the correlation. And you say, oh, look, the disease looks like it's exactly the pattern of maturation or the pattern of expression of a certain gene. And this work by Justine Hansen and Bratislav Misic related the ENIGMA disease maps with all sorts of brain maps that had been collected on connectivity, dopamine receptors, serotonin receptors. I mean, you could say, well, I think wherever the dopamine receptors are, maybe that's the place where people are losing cells. And so this is early days yet. You'll see that they're trying to make a list of disorders along the bottom, and then their favorite maps of laminar differentiation, receptors. I think we should suspend judgment for a little bit. I mean, it's exploratory, but I mean, it's an interesting idea to go from disorders at the bottom to brain maps. And there may be some cellular tag or gene that's being expressed that has a very high correlation with what we see in the images. So if you want to do this, there's something called the ENIGMA toolbox. You can relate your disease maps to all sorts of other multimodal data on brain connectivity, molecular maps and so on. And it's quite good. It's developed by Sarah Lariviere in Montreal. And so what are the new directions? We talked a lot about brain structure. There's three of them that ENIGMA is exploring at the moment. So one of them is AI and deep learning. So we heard Andy Greenshaw talk about AI in psychiatry. Another area is to look more at brain connectivity. We know that the disorders are typically disorders of the way that parts of the brain communicate with each other, rather than any one region being the culprit. And then also there's newer groups on TMS, neurofeedback, meditation. People, as you know, might use mindfulness as an approach to treat chronic pain, PTSD, MDD to some extent. And AI models are very different. So rather than FreeSurfer or these packages that analyze images, you just throw all the data in, genes, images, maybe you bought a watch. Andy Greenshaw talked about real-time health data. And then you train a neural network to come up with a decision, either the patient's diagnosis or their prognosis or the best treatment. And with enough data, you optimize the weights of this function, just a mathematical function, that combines these variables in such a way that you do better and better on these tasks. And so there are CNNs, there are all sorts of cool methods. And these actually work very well in neurology. So in Alzheimer's disease, you can train a CNN or a convolutional neural network to detect Alzheimer's disease with 91% accuracy. In fact, there's a lot of competitions. So people are developing deep learning methods to detect disease, and the accuracy goes up to about 90% with more training data. Once you have about 2,000 MRIs, at least for Alzheimer's, you can detect this pretty well. And there's a lot of competitions. And so there's video and synthetic MRI pre-training vision architectures for neuroimaging. So what we want to do in ENIGMA is try these in psychiatry. It'd be very interesting to see if these would work in psychiatry. The other one is to look at the connectivity. So we heard about diffusion MRI yesterday. There's a lot of software. There's some cool software in Boston by Anastasia Yandicki and colleagues. But this is different. It's DIPI. It's a diffusion imaging toolbox that we use in ENIGMA. This is Bram Chandio. She's a postdoc who wrote Bundle Analytics. And it will essentially break the tracts of the brain up into segmented bundles. You can chop them up like that. And then you can say at each point along the tract, tell me where the diffusion metric of interest, maybe it's free water, or maybe in this case it's fractional anisotropy. Tell me where that is different, in this case bipolar disorder versus controls. And that's just starting. People are beginning to see effects in the cerebellum. They're seeing expected effects in bipolar, where the fine scale maps of the frontal limbic microstructure are showing anomalies in bipolar disorder. Now, all this business of site harmonization comes up with these maps as well. So one of the things that Ram Chandio, she's a postdoc with us, is doing is adjusting the data with a scanning protocol at each location along the tracts. Now, you could say, well, isn't the whole image affected the same way? But it's not. So if you have a diffusion MRI with large voxels, it won't pick up fiber crossing very well. And so it's not the case that the same adjustment can be applied everywhere in the scan. So there's a lot of mathematical research on that. And these maps are cool. I mean, this is actually a study we're doing in India of tract measures in different systems. And this is the effect of Alzheimer's disease on the left and mild cognitive impairment. Again, greater in the temporal lobe, which is where we said at the beginning that the deficits arise early in the disease. The last thing that's kind of fun and it's new is to look at air quality. So Lauren Salmon and Megan Herting and J.C. Chen said, well, you're missing the point. ENIGMA doesn't look at the environment. And in fact, a lot of the disorders that we get come from the environment, or at least are potentiated by environmental hazards. So I bought this gadget. I've been telling you about this. So any of you could buy the thing on the right. And just to reassure you, here in Khmer, the particulate matter is 0. It's the first place that I live in Los Angeles, so it's horrible there. In fact, this was in Abu Dhabi. That picture over there is, where was it? I flew through Doha. And there's a nice sunset, but look at the amount of pollution. The pollution was, well, there it is. The reading is pretty high. And the other reading is radon. My daughter said, don't take this on the plane, but I did anyway. So you can map it. I mean, we could go around with these gadgets and make maps of pollution. I mean, look at where the pollution is bad. And I'm not saying it's good in the United States, where I am. In Los Angeles, it's one of the worst in the world for pollution. But pollution with particulate matter, different organic compounds, there might be dust. I mean, they have slightly different distributions. And it may as well be measured either through geocoding, which is you estimate it from the population data, or with a device that you can have the patients take home with them. So that's kind of fun. What are the lessons that have been learned by working internationally in these projects? So I think, as you all are seeing, global participation is important for science. There's activities in ENIGMA in 45 countries. And it's very politically beneficial when projects are led from different countries. So people will often say, and you know this, oh, have you noticed this disease risk factor? And they may have observed it in their own patients, and then other people can have a look at that. So people talk about the power and efficiency in the computing, but there is also more expertise. If you write a paper with other colleagues, I mean, their expertise is often going to guide you in a better direction. So we have an India ENIGMA initiative, which is kind of interesting, lifespan charts and risk factors in Bangalore. What is different in ENIGMA about your risk for dementia? Well, one of the things there's things that are better and things that are worse. So in ENIGMA, in India, the sanitation is poor. The access to healthcare is poor on average. The social support and the family support in India, if you're an elderly person, is excellent. It's much, much better than in the US, where you're sort of abandoned. So there's interest in which of these factors cause the brain aging to go differently in India. There's a similar project in Thailand. So the Red Cross in Thailand leads the ENIGMA HIV group. So women who contract HIV in Thailand, if they give birth, the baby often has HIV infection passed on from the mother. And there's a lot of interest in this group in the Thai Red Cross in making growth charts for children that are treated with antiretroviral medication for HIV. We did have a really active network in Russia. In fact, it was a really strong partnership with Russia. I think it's been difficult for political reasons, but I think that will probably improve. There's a very strong ENIGMA network in Iran. In fact, they have an imaging consortium across Iran, but for political reasons, because we're in the US, it's very difficult to publish with people in Iran. And here's our friends. So many of you know Dr. Ali Safet Gönül and Aslihan and Yiğit and Cemre, all of the people in the SOCAT lab in Izmir. And and there's Shadash as well. They have been really doing pioneering work in major depression, bipolar, many other conditions using imaging, and we're really, really happy to be working with them, leading some of the really cool projects that you see here. And by and large, you say, well, are the brain differences very different in different countries? I think the initial message is that they're not really very different in different countries. I mean, in Japan and the rest of it, the brain differences tend to be rather similar. Maybe socioeconomic status is a factor. So it's difficult to study because socioeconomic status in the United States is correlated with body mass index, lower body mass index, higher wealth, and it's correlated with educational attainment, higher socioeconomic status, higher educational attainment. And whenever three variables are highly correlated, you can never disentangle them. So you have to go to a place where wealth is not correlated with education, and where wealth is not correlated with body mass index, which is much weaker in Qatar. So you can imagine that pooling data from different countries where the risk factors are not correlated will help you disentangle the effects of these multiple factors. So just in summary, ENIGMA coordinates worldwide neuroimaging studies of 33 disorders across 45 countries. The brain disease signatures are consistent worldwide, but they change with duration of illness and sometimes with treatment. The genetic studies reveal over 500 loci that affect the brain. Some affect the speed of brain development and aging. Environmental studies are also underway. And then the newest studies are beginning to look at functional MRI. Now, it could be that functional MRI is the best way to look at these psychiatric conditions. It probably is. I mean, if you go to the brain mapping meeting every year, most papers are using functional MRI. They don't behave like we're all radiologists looking at structural and diffusion measures. So the newest studies piloting in ENIGMA are using fMRI to study interventions, things like TMS, other forms of neuromodulation, neurofeedback, even meditation. And if you want to join in, if you're scanning patients, you're very welcome to join in. There's a YouTube channel that describes some of the analyses that are going on, or you can just contact us. You're very welcome to look at ENIGMA's project, see if there's anything that you'd like to get involved with. And a huge thanks goes to all the people that are leading and participating these efforts. There's even groups in Mongolia that are scanning. So in Mongolia, the pollution is really bad. It's a very, very nice country, but in the winter it's minus 40 degrees and they live in yurts, and the little babies have a lot of coal smoke affecting their respiration. So there's a lot of interest in looking around the world at different risk factors, because some of them might be important to us and they're not the most important ones for people in other parts of the world. So this is funded primarily by the NIH, but also by some private and European and Asian agencies. And this is sort of funny. The quote, very old quote here, that it's actually from Aristotle, that individually we contribute little to the quest for truth, but working together the whole vast world of science. I don't think Aristotle said science. I think he said knowledge. The whole vast world of knowledge is within our reach. And this is in the early works of Aristotle. Oh, I wanted to thank the translator. So there's something interesting about this meeting, very interesting about it. The translators at the back are doing simultaneous translation. And I wanted to say, Buddha situche cheviren teshimana teshikular. Very kind of you to listen to everything we say and within 1 or 2 seconds produce a Turkish or English translation. So I have an immense admiration for what you've been able to do. So thanks very much. And that's it. Thanks for your patience. Appreciate it.